Hi, welcome to Biology Tunkit, and today we'll be discussing a very important and very interesting topic, which is known as linkage. In order to understand what linkage is, let us understand what are the genes. Genes, you might know, genes are the unit of inheritance. Genes are the unit of inheritance, meaning that all the characteristics that develops in a particular species came from the gene. Now we take two genes, naming them as A and B, and as we all know that genes are present on chromosomes. So if these two particular genes are present on a single chromosome or the same chromosome, single chromosome, and linkage between these particular two genes are present, then according to linkage, meaning that when these particular chromosomes will be assorted from parents to offsprings, then there will be a tendency of both the genes to assort together, meaning that during assortment, if I briefly tell you about Mendelism, then you know there is a law of independent assortment that have been constructed by Mendel, and according to that law, that genes usually segregate from each other during the formation of gamete. Okay, and now if linkage is present, then these particular two genes during forming gametes, if linkage is present between these particular two genes, then they will have a tendency to assort together into offspring. Okay, so this particular phenomenon was noticed by a scientist named as Beston. He noticed, how he noticed? Because he was performing a dihybrid cross with pea plant, and he noticed some deviations that from the Mendel's law. So based on that, he concluded simple two terms, that genes on a particular chromosome can have arrangement of two types, which he coined as coupling and repulsion. So what happens during coupling and repulsion? Let me quickly draw this. So I have drawn two chromosomes. Now let us consider us that these particular two chromosomes are homologous chromosomes, meaning that they have a particular homology, meaning that gene arrangement in these two particular chromosomes are equal or similar. Okay, so in coupling, what have been told that genes will be present, a dominant form of a gene will present on a single chromosome. That is, if A is a gene, then dominant form of that particular gene will be present on a single chromosome, and the recessive ones will be present on the other pair of the homologous chromosome. Okay, so this particular fashion is known as coupling. So if I write it as this, then B. Okay, so this particular arrangement is known as coupling. And in case of repulsion, what happens is that a single dominant gene presents on one, and the other dominant form of the gene remains on the other pair of the homologous chromosome, and the rest is just like opposite, small b, and here is capital B. So in case of repulsion, what happens? So this particular arrangement happens during repulsion. Okay, I hope it is clear now. So now comes the interesting part, which includes that the entry of Morgan. And I forgot to tell you that the linkage term is coined by Morgan, because previously when we saw that, based on have noticed some deviations in the law of independent assortment, he cannot concluded what is a particular phenomenon, but he have observed, and he have hypothesized that these particular two arrangements can be present in a particular gene. So Morgan conducted an experiment on the fruit fly, the fruit fly, where he took two characteristics or two traits. Two characteristics means he performed a dihybrid cross. So characteristics were, a fruit fly usually have a gray color which is dominant, and a recessive one which is black, and another character was the size of the wing. So usually have a long, which is denoted as capital N, and a recessive one which is known as short L. Okay. So now the phenotypes of the two particular flies were capital G, capital L, small g, small l, small l. The phenotype includes gray body color plus long wing, and black body color plus short wing. These are the characteristics of the parents. Okay, these are the characteristics of the parents. And now when he performed a cross between them, he got, first the gamete formation will take place. So from the first one, I'm naming it 1 and 2. From the first one, what gametes will be produced? Gametes will be G and L. These are the gametes will be produced. And from the second one, small g and small l. These gametes will be produced. And now hybridization takes place. And lastly, the genotype will be G, capital G, small g, capital L, small e. This is the filial or the progeny, F1 generation. Okay. So now suppose that in these particular two gametes, there are two genes. One is G, another one is L. And in case of this particular gamete, one is small g, another is small l. And now if between these two particular genes, if linkage is present, then there will be a probability that these particular two genes will accumulate into the progeny together, and in contrast to that, this also can happen. Okay. So now what Morgan did was he performed a test cross. And in test cross, we usually cross the filial one or the progeny with the homozygous recessive one. So he made a cross between capital G, small g, capital L, small l, that is the filial or the progeny one, with the small g, small l, small l, with the recessive homozygous one. Okay. So from this particular, there will be 4 types of gametes can be formed. One is capital G, capital L, capital G, small g, capital L, small g, and small l. And from this particular, only one type of gamete can be formed, G and L. Okay, this is a single type of gamete. So now if you remember in test cross from that of Mendel, and you probably know that if the heterozygous is present, then there is a phenotypic ratio of 1:1. If I would have performed a monohybrid cross, but since I have performed a dihybrid cross, my expected result would be 1:1:2. This would be my expected result, because I have considered two particular genes. For one gene I would have this particular one, but now I have two genes, so I have this particular expected result. This is my expected result. But the results that came out was astonishing, because the results came out as something like this. this type of result came out. So why this particular result came out? Because of that single particular phenomenon which we have learned throughout the class is linkage. Because there is linkage between capital G and capital L, or I say there is a coupling, or I say there is a coupling is present between particular genes. That is why the progenies which have yielded, or the progeny which have been produced, have characteristics like that, majority characteristics of like that of parents, which includes majority of the offsprings have gray color plus long wing, and black color plus short wing. And there are very minute numbers of recombinant have been produced. So we can neglify that. So majority, if there is linkage will be present, linkage is present between two genes, then what should we expect? We should expect that the results that we are going to achieve will have more characteristics. Will have more frequency of the characteristics just like that of parents, because we started with parents with what? With gray and long wing, here is it is, and black and short wing. And now the results which have yielded have gray color and long wing, and black color and short wing. So this is a complete deviation from that of Mendelian theory. Okay, so this is the case of coupling. So what if repulsion is present? So repulsion, in case of repulsion, same kind of results would be yielded. This particular results would be yielded. So I hope this is clear. If not, do ask me question in the comment box and I will be happy to help you. Okay, bye.